The rich profusion thee confounds, my love, of flowers spread athwart the garden. Ay, name upon name assails thy ears, and each more barbarous sounding than the one before. Like unto each the form, yet none alike, and so the choir hints a secret law, a sacred mystery. For those of you who are not familiar with the absolute giants of world literature, take a moment to Google up Johann Wolfgang von Goethe, or read his Wikipedia entry. Scholar, statesman, poet, and author, he's most remembered for his literary contributions, like the drama Faust, based on a legend that's familiar to us all, of a man who forms a pact with the devil, or Mephistopheles, who will serve Faust on earth, and then Faust will serve the bidding of the devil in hell forever after. The story has been told over and over, but Goethe's version will always stand out as the greatest. Personally, my second favorite version is the one with Sammy Davis Jr. and Christopher Lee. Yes, that is Count Dooku in his younger days playing Satan. Getting back to Goethe now, if you consider carefully the lines in this poem, and this is just the start of a much longer work, you might be saying, yes, this is exactly right. I totally understand this. Name after name, each one harder than the one before. Orobank KCE, Jesneri ACE. Where is he getting these words? It's like he's speaking in a different language. The choir hinting a secret law, a sacred mystery. Well, Goethe understood the baffling nature of botanical names over 200 years ago. But the real reason I'm opening this video with a quote from this particular work of von Goethe is that the Metamorphosis of Plants, published in 1790, had such rich detail in describing the relationship between leaves and floral organs. It's so stunning and accurate that you might wonder if you might have gotten some inside information from a time traveler from 1990, or maybe even the devil himself. And by the way, this work, The Metamorphosis of Plants, it only begins with a poem. Most of the book is just in regular prose. I'm not sure when, but sometime between then and now, we've lost this practice of introducing one's research with dozens of lines of poetry. Now honestly, I've only read bits and pieces of Goethe's analysis of plant reproductive anatomy. Really, its greater value is to historians of science rather than practicing scientists. But reading these bits and pieces, I hear in my mind some of the same things that I present to students today as modern lessons in developmental evolutionary genetics. And it seems just so strange that these ideas and concepts, they've been around for so long, how could they still be current and modern today? But this is because we've only recently come to understand the genetics underlying the transformations of which Goethe speaks, such as leaves transforming into sepals and petals, and stamens, and carpels as well. Now, I've promised you a couple of times that we'd be doing more on flower development and the secrets that it may hold in relation to the rapid angiosperm radiation. In biology classes, this general topic of development is big and important, and we always spend quite a bit of time on it, though it's usually all about animal development, and in particular the changes that occur between zygote, that is a fertilized egg, as it develops into a young animal. And sometimes people forget that plants are even there, and this whole topic of development is treated as an animals-only phenomenon. But we're not going to do that. Plant development, and in particular the development and specification of floral structure, may turn out to have a central significance in our understanding of that all-important question of how angiosperms have been able to undergo such a radical and rapid evolutionary diversification. But we're going to start with conifers and gymnosperms before returning back to flowering plants later. We've seen how conifers make use of cones. The female cone bears megasporophylls on which the megagametophytes develop. Male cones, which you're looking at here, bear microsporophylls that bear massive quantities of pollen, which are the mature microgametophytes. Pollen travels by wind from tree to tree, from the tree where it's produced to the next tree where the pollen may fertilize the megagametophytes in the ovulate cones, thus making a seed. Now the result is a life history in which the full alternation of generations can occur without any of that helpless, free-living gametophyte stage like what you see in the ferns. Instead, the gametophytic stages complete their development under the protection of the big sporophytic parent plant, And this has afforded pine trees and seed plants in general a lot of success in drier habitats. Now the cones are central to this success, and so the question we start out with regards the making of the cones, how a pine tree uses genes to trigger the production of cones, which are, as we will see, just developmental variations of the vegetative boughs of the pine tree. I'm calling them boughs, but that's kind of an old school poetic term, like when the bough breaks, the cradle will fall. What I mean is the vegetative branch that produces both leaves and lateral branches. Draw you a picture here. Here's some lateral branches. Put some leaves on the lateral branches. Okay, you get the idea. Now looking again at this cluster of male cones near the tip of this branch. Okay. Right here at the very tip, there's non-reproductive or vegetative growth, more needles, and a continuation of the growth of the green bough. The cones that you're seeing over here are really modified lateral branches that were triggered into developing as cones by the gene leafy. When this gene is active, lateral branches develop as cones. When the gene gets turned off, then growth as a vegetative branch resumes. So what happens when leafy is turned on? Let's pretend we're turning leafy on right here, in this area right here, right? So leafy is going to be turned on in that region of our little branch. What's going to happen is that these lateral stems are going to develop as the central axes of male cones, and instead of needles, the leaves will develop as microsporophylls, which are the scales or bracts that make up most of the male cone, and they're also the parts bearing the large pollen sacs. So that's my attempt to represent a single cone, a male cone. Each one of these little bracts, each one of those little things that looks like that, is actually a microsporophyll that has hundreds of thousands of pollen grains inside. And so when the staminate cone's open, when the male cone's open, lots and lots of pollen is going to come out. You can see that this spring. Now in the case of the female cone, the structure is a bit different. When they first form, they look like this. And so after a year, those same female cones will have grown in size, although they haven't yet fully matured. And you can see that the vegetative growth is going to continue on through the middle. And you have a continuation of the vegetative growth out here, and the two female cones are much, much bigger, but they're still not mature yet. Okay? And ultimately, maybe after 2 years, maybe after more than 2 years, you end up with mature pine cones, which when they open, these bracts will release seeds. Okay, so these main seed-bearing scales, structure right here, seed-bearing scales, sometimes called the ovuliferous scales of a pine cone, of a female pine cone. They're not leaves, they're not modified leaves. Remember, the male cone, the scales are actually modified leaves. In the case of the ovuliferous scales or bracts on a female cone, those are actually lateral stems. Okay, so it's kind of like the main stem is the main branch, and then you've got a lateral stem from which you've got leaves. There's a couple of modified leaves on the bottom of this, and the megagametophytes are born at the base of these lateral branches, or these ovuliferous bracts. Now, as I said before, leafy is the trigger for initiating the development of female cones, and I must confess my ignorance of how it's determined whether the lateral branches develop as a male or a female. I've heard a couple of ideas, but nothing definitive. But it is clear that the signal from an active leafy gene is what's required in order for the plant to initiate development of a reproductive structure. A cone, in the case of the pine tree, or in the case of an angiosperm, a flower. Now, in the absence of the signal, the stem just carries on with making more and more green boughs, which is kind of like the default growth pattern for all of the above ground part of the plant. Now, Goethe mentions pines a couple of times in his book, but mostly he talks about flowering plants. And through observations of the variations among the different types of flowers, he's able to conclude that while leaves, sepals, petals, stamens, and carpels are clearly distinct structures, they're also somewhat free to transform from one structure into another. Now, this is not very different from the observations that led us to the ABC model of flower development. One particular plant species, the cress, Arabidopsis thaliana, was the model study organism for much of the research leading to our present state of understanding. Some of the early studies with RAB, as it's sometimes called, noticed that mutations affecting the flowers tended to affect the development of sepals and petals together, or stamens and petals together, or stamens and carpels together. You never had a mutation that only affected one of the floral parts without something else. Now, one of the easier ways to visualize the ABC model is to think of a flower in a stretched out sort of way, with the different floral parts coming off of its central axis in a linear sequence from the base to the tip. Here, let me bring this slide back up. We've already seen this with a male cone. If this is a central axis, we've got lots of modified leaves. In this case, they're all microsporophylls. And I want you to think of a central axis again, but in this case, it'll be a regular flower where you've got sepals coming off from the base, then you've got petals, then you've got stamens, and then you've got carpels, which fuse into a pistil at the tip of the axis. Okay, so I'm going to try to draw this out for you. Okay, so there's the central axis, and I've drawn the leaf primordia here. These are the things that are going to be growing either into leaves or sepals or petals, etc. Okay, so in a nutshell, you've got the expression of three classes of genes, A, B, and C, and they also follow a linear base-to-tip sequence. The A genes are expressed in the bottommost portion of the axis, and then you've got the B gene whose expression overlaps somewhat with the A, and then you've got the C gene whose expression goes all the way out to the tip, and it doesn't overlap with the A, but it does overlap with the B. And once you have this gradient of signals established, then the development of the leaf primordia just follows a set of rules. For example, if there's only A, which is exactly what you have at the base, the leaves develop into sepals. Further up on the axis, you have both A and B, and this causes the leaf primordia to develop into petals. Those are supposed to be petals. If you go even farther up on the axis, A is no longer present, but you have a combination of B and C, and this is what makes the leaf primordia develop into stamens. And at the tip of the axis, you have only C, and this is what causes the leaf primordia to develop into carpels, which then fuse to form a pistil complete with a style. Then you've got a little puffy stigma coming out from the very tip. And so if you could visualize this and try to scrunch things down into a much shorter axis, you can actually sort of see how this is going to give rise to a flower. Here is a real Arabidopsis thaliana wild-type flower, and you've got the sepals, base, petals, and then outside of that you've got the stamens. And here you can see the stigma popping out from the middle of the stamens. Okay. And this is obviously an oversimplification, and I want to be clear that while this model works for Arabidopsis, other plants, different species of plants, may use their A, B, and C genes in a somewhat different way. But this actually becomes important later in our discussion. So now that you have the fundamental concept underlying the ABC model of flower development, I'm going to narrow in on just the class B genes, the ones required for the development of both petals and stamens. By knowing the sequence of these genes individually, and also having the complete genome of Arabidopsis, we can see that there are actually many copies of the B class genes falling into two main subgroups. I call them pistillata, or the PI group, and apetala 3, or the AP3 group. Both pistillata and AP3 are required in order to carry out the function of specifying normal petal and stamen development. But both of these are present in multiple copy. I guess the first question is to ask, why are there so many extra copies? Well, of course, it's the result of this plant's history of whole genome duplications. So one of the surprising findings from the Arabidopsis complete genome is that these little plants carry a boatload of copies of this basic gene that in our oversimplified picture of flower development is just the petal/stamen gene. And it's not just the B class genes, it's not just pistillata and AP3. All of the genes that are associated with flower development are present in multiple copy. What this suggests is that these floral identity genes are not selectively neutral, that the plants have maintained the extra copies of these genes because they gave them an advantage over those plants in which the extra copies were pruned away. Remember that predictable reduction in genome size following a polyploidization event. So here's where we start to inch our way back towards the Asterid-focused discussion that we were in before getting sidetracked by Goethe in this issue of flower development. So in 2009, Vienna et al. Reported this massive study of the many PI and AP3 genes in dozens of species of asterids, tracking their pattern of development. Okay, so here's a list of all the different species that they used. And the genes are over here. These are basically the copies of PI and AP3 in a closely related group of genes, TM6. Scroll all the way down, and basically what they did was they set things up, and they're able to detect the expression patterns of these genes in different parts of the flower. And basically what these results show is that while these B-class genes are essentially the same, the different copies, they differ in the how, the where, and the when for their deployment in floral development. Okay. And this is true not only between the species that we're looking at, but also when you look at the various copies of the gene that are there in the genome of a single species. And this is exciting because it illustrates that kind of specialization we talked about in the earlier video. I think it was the 10th video in the series. The idea that genes start off as identical in the aftermath of a polyploidization event, and then over time they become more specialized. And this is basically a luxury that's made possible by virtue of these extra copies of the genes. And I'm hoping that you might even remember that this was one of those situations in which having extra copies of genes is actually advantageous for the plants. Okay, this also opens up a potentially important mechanism underlying the rapid evolution of floral morphology. If you are a plant, how do you go about making your flowers evolve? Well, with slight changes in the timing and placement of the growth of your sepals, petals, stamens, and carpels. And how does having extra copies of the floral identity genes facilitate this? Well, the general idea is that you can keep one copy working in the old way, to make sure that you basically have a functional flower. And the mutations to the other copies allows the plant to freelance, exploring slightly different floral shapes and patterns that might allow the plant to evolve, for example, in sync with the pollinator species. Okay, so the picture that emerges here is one in which genome duplications, which until not that long ago were thought to be of minor importance in evolution, they've become key to understanding some of those big questions that were there right at the core of Darwin's abominable mystery. The how flowering plants did it questions when it came to their extremely rapid evolution and diversification in the mid-Cretaceous. Now returning to our survey, we're ready to take on the other half of the superasterids. And basically last time we talked about these guys over here as being the Lamiid half of the superasterids, and we have the other half of the superasterids that we haven't detailed yet, right? The campanulids. Okay, and I was thinking about redrawing a new tree for this video, but we really have only 3 orders here. Okay, in the campanulid group, we're only going to talk about 3 orders, and I'm just going to put them right here. I'm going to snake them into this diagram. So one is going to be the Asterid, one is going to be the Aquifoliales, and then the third one is going to be the Apiales. Yeah, all 3 of them start with A. The Asterid is the order that gives us the Asteraceae, or the sunflower family. It's going to be very familiar to you. It's also a huge family with more than 23,600 species, more than all the ferns and conifers put together. Daisies, zinnias, marigolds, chrysanthemums, dahlias, echinacea, and of course sunflowers are all asters. Many kinds of foods besides sunflower seeds that are from this family include lettuce, artichokes, radicchio. If you like chicory, all kinds of chicory are also in the Asteraceae. But the most crucial bit of instruction that I want to give you regarding this family has to do with the structure of the pseudanthium, which basically means fake flower. And that's really not true either, because flowers are in fact there. Let's take a look. Okay, daisy. Everybody recognizes this. But if you think that what you're looking at here is one flower, then you definitely need this botanical lesson. Okay? What we're actually looking at here is an inflorescence comprised of dozens of flowers that are clustered together. Okay, so let's do a diagram. What I want you to first visualize is just a regular flower. And so this might start off with a little calyx. These are the sepals. And this is kind of like a fused tubular flower, kind of like what we saw in the Solanaceae, right? And normally you would say, well, the next things to come out from this flower would be the petals. And yeah, that would normally be the case, but let's say this is a very reduced flower, so we don't have the petals. And all we have are the interior parts of the flower. So maybe there's going to be a stamen coming out of here, that's an anther and a stamen. Or to make this actually fit our picture, let's visualize there's just a little stigma coming out. Okay, so there's a stigma, there's a style, and the stigma is the sticky part, the thing that's going to attach to the pollen grains. Okay, so we're dealing with a very, very reduced flower. It's a tiny flower. And if we look at it head on, it might look something like this. Okay, so that might be the fused calyx. And there's a stigma that's coming out. Okay, so it doesn't look like a whole lot head on. Now these are really tiny, and I want you to put a whole bunch of these things together. So I'm going to make this multiply. And so you could take dozens to hundreds of these little tiny flowers and pack them together into a central disc. And then you have the middle part of a daisy. These are called the disc flowers. Okay. And now I want you to visualize another kind of flower, basically very similar to a disc flower, only this one is going to have a corolla. So basically we're starting off with the same calyx, and the corolla in this case is going to be elongated. It's actually a tube that's open on one side, so you've got the strap-like structure. It's actually made of 5 petals that are fused together into a single strap. Okay? And again, you'll probably have some type of floral structure, maybe a stigma coming out over here, maybe an anther coming out over here, and maybe a stigma as well. And so this would be the other kind of flower. This would be the ray flower, in contrast to the disc flower. And so you could visualize these ray flowers aligned all the way around the outside. And so you get the idea. Lots of disc flowers forming the center, ray flowers forming these things around the outside. These are not petals, right? Technically they are petals, but they're fused into corollas. Each one of these is the fusion of 5 petals forming the corolla of a ray flower. Now with this in mind, we can take another look at that daisy. And now looking at this, I'm hoping you could see. I mean, let's zoom in a little bit closer, right? If we zoom in, yeah, you can actually see that, can't you? For example, this whole structure here is a fused corolla of one ray flower. You can actually see, it looks like the stigma there. Over here you could see a disc flower. Looks like there's an anther popping out from the middle there. And so even though you might have up until this moment thought of this daisy as a single structure, from here on forever after, this is no longer a flower. It's an inflorescence, or a pseudanthium, comprised of many flowers. Okay. This combination of ray flowers and disc flowers is where we get the old name for this family, the Asteraceae. We used to call them the compositae. And in fact, we still refer to these pseudanthia as composite flowers. Okay? Now, we haven't yet unraveled the developmental genetics underlying the sequence that leads to such a highly derived floral morphology. But the huge number of species in the Asteraceae, remember we've got 23,000 some hundred species of asters, that huge diversity could very well be related to the increased potential for diverse form and flower structure. So in this sense, in the context of what we've gone through so far, it's not surprising at all to learn that the Asteraceae, this family, had its own family-specific genome duplication. And when we analyze this genome duplication's age, it seems to have coincided almost perfectly with the time of the radiation of the Asteraceae. Okay, now getting back to our tree. We've talked about the Asteraceae, and now we'll be talking about the Campanulaceae, or the bellflower family. It's the only other family we'll mention within the order Asterales. It's a pretty large family, though nowhere near as large and diverse as the Asteraceae, and we encounter them on a regular basis, mostly as ornamental plants. Here I'll show you a couple of pictures. So over here on the left, we've got the pretty bell-shaped campanula, or the bellflower. And on the right, we've got lobelia, which is characterized by tubular flowers with lips. Okay, that's the Campanulaceae. Now moving on to our next order, the Aquifoliales. The Aquifoliales is for us just a single genus, Ilex, which is in the Aquifoliaceae. So both of these are in the genus Ilex. We mostly know ilex from the holiday decorations you see everywhere around Christmas time. Um, European holly, ilex is kind of like a symbol for the holidays. But there may be some of you who are familiar with a drink called yerba mate, which comes from Ilex paraguayensis. Um, it's native to South America, and it's very, very popular in Argentina. So that's Ilex, including holly and yerba mate. Now the Apiales is another large and familiar order of plants which contains some of my favorite veggies and herbs. The main family in the Apiales is the Apiaceae, which is characterized by umbel-shaped inflorescences. So you probably don't know about umbels, so the best thing for me to do is just to show you what an umbel looks like. Okay, so this is kind of like a dried out head of the parsley plant that I let go to seed. And the shape of this inflorescence has stalks emanating up from a central hub, arcing upward to a flattened top, and that's an umbel. And besides parsley, members of the Apiaceae that you should know about are carrots, celery, fennel, dill, what else? Cilantro. Okay. Apiaceae. Our next family in the Apiales is going to be the Aureliaceae, which is the family that includes the Hedera or ivy plant, as well as Panax, a ginseng plant used in Asian medicine. And that's the Aureliaceae. Now one last family is the Pittosporaceae. Now the Pittosporaceae is the family of one of the few trees that I truly dislike. Okay. There are certain plant species that become recognized by the horticultural community as being easy to maintain or good for some crazy reason. And the next thing you know, they get planted everywhere. And then we're looking at the same trees in the planters of parking lots for the next 30 to 50 years. Now this plant, you've seen it many, many times. Pittosporum tobira, Japanese cheesewood. This is unquestionably one of those plants. The Pittosporaceae really should have been a skipped family in our survey. But since it's getting to the point where we see Pittosporum every single day in our urban and suburban forests, I'm just forced to include it. Now, to be fair to the family, there are plenty of other species in the Pittosporaceae that are interesting plants. I mean, legitimately kind of cool plants. I just kind of reject the way our city planners have given us such a huge amount of blandness to look at. You know, Pittosporum and boxwood and Indian hawthorn, the same three plants over and over again. Okay. Probably shouldn't have got started on that. And that pretty much does it for the Asterids. Putting together the three A orders, Apiales, Aquifoliales, and Asterales, and this group, the campanulids, together with the four orders we talked about in the previous video. Okay. Now, I knew that we had relatively little material for this survey here, which is why I inserted the discussion of the floral developmental genetics before getting into the Asterid II groups. And the timing was actually appropriate, since some of the most recent interesting work done on diversification and evolution that are using the products of genome duplication have been done using plants in the Asterid lineage, and Vienna's study is just an example of this. Okay. Moreover, the Asteraceae in particular, remember those composite flowers, that's probably our premier example of diversity of form that we find within the whole plant kingdom.